Alright, welcome everyone. It's the first time I've used this screen recording function, so forgive me if it's a little bit rough. But I thought it might be helpful to just have a video that you can come back to in case you want an extra explanation of Trp Operon, because I know that it is a part that people tend to find difficult. Okay, I think a lot of that comes from some of the new words you might come across, which makes it seem more complicated than it is. Okay, so what is Trp Operon? Okay, well, TRP is essentially just the shorthand for the amino acid tryptophan. Okay, it's a really important amino acid that's used in a whole heap of different things, and it's something that the bacteria needs to make. Okay, it's hard to find it, so it needs to make it. Operon essentially just means a group of genes that code for a particular thing. Okay, there are all sorts of different operons. So in this case, this is a TRP operon. It's a group of genes that are going to end up making TRP. Okay, it's as simple as that. So when we look down into this drawing below, which are from Khan Academy, we see this TRPE, TRPD, TRPC, TRPB, TRPA. Okay, all that is 5 small genes that code for enzymes that are going to end up creating tryptophan. So these are all kind of precursors that are going to end up making tryptophan. Okay, you don't need to know those in detail. Just if you see a picture, that's what those are. Okay, so that's the operon. Now, you will see a couple of similar words. So we see the word operator. That is essentially a section of the nucleotides where a repressor is going to bind. Okay, and we're going to talk about that when we talk about how the cells manage how much tryptophan they make. We see the word promoter. That's essentially the same as a promoter anywhere else. It's the sequence of nucleotides where our RNA polymerase is going to bind in order to start that transcription and translation process. Okay, it's as simple as that. So let's have a look at the 2 scenarios that we're going to talk about with this sort of primary gene regulation strategy. Okay, so we've got 2 different situations. We've got a high tryptophan situation and a low tryptophan situation. Okay, so obviously we need tryptophan when there's not much around. Okay, it'd be silly to make tryptophan if we don't need to. Okay, so that energy saving is a massive part of why bacteria have all these different ways to regulate it. Okay, it's a massive waste of energy if we're making tryptophan when we already have plenty. Okay, so we talked about the operator before. It's a sequence just before the genes where a molecule, a repressor, can bind to. Okay, so this red thing here is the TRP repressor. Okay, now the kicker is that the TRP repressor only binds onto this operator if it has a particular shape. Okay, and it gets that shape by binding with tryptophan. Okay, so how amazing is that? The presence of tryptophan, if it's a high tryptophan environment, actually changes the shape of the repressor so that it can bind onto the operator and stop this RNA polymerase from joining on and making those genes that create tryptophan. Okay, we don't need to create tryptophan because there's always some present, okay, or there's already some present. And it's the presence of that tryptophan that actually activates the repressor. Okay, now let's say we use up all our tryptophan. Okay, we've used it in our protein production, and now suddenly we don't have any tryptophan. Okay, so now it's a low tryptophan environment. Well, now the tryptophan that was previously joining onto that repressor and allowing it to bind onto the operator is suddenly not there anymore. Okay, so we take that away, and now suddenly our repressor can't bind to our operator. So now we see it floating just in the cell or in the bacteria. And now we can see that RNA polymerase can bind to the promoter and start that transcription process. Okay, and if you think that next step forward, this sequence is going to be transcribed, translated into proteins that are going to end up being the tryptophan. And then suddenly we're going to go back into a high tryptophan environment where it's going to then repress the TRP transcription. Okay, so we're going to reactivate this repressor, which is going to bind to that operator, and now we're in this situation until it gets low again, and then you can see it's basically a homeostatic loop. Okay, a a high tryptophan environment stimulates the repressor, a low tryptophan environment deactivates it so we can make tryptophan, and it goes around in a circle. Okay, the next video will be on attenuation.